Okay, can somebody confirm you can hear me?
I can hear you.
All right. So let me state that I got a new worksheet for lecture 8B up. And here it's here, and it just has whatever was initially on 8B, and then it's added more questions to it. I also need to talk about, before I share my screen, I didn't talk about the grading. Last week, I got several things graded, and I don't want to share my screen because I want to call up the grade book so then I can remember what the heck I did. I believe I got another extra credit study group assignment graded, but only one. And I'll try and get those worked on further. I got worksheet for lecture 6 graded. I got worksheet for lab 3 graded the second time. And I got worksheet for lab 8 graded the first time. Now, worksheet to lab 8, you may, if you did not get 100%, you may resubmit the lab by this Saturday, and that will be for full credit. Any question about the grading? I have not looked at the infectious disease research project. Let me remind you that this project will be checked for plagiarism, and you can just take a look at the color code. If the color code is green, everything is good. If the color code is yellow, I'll go take a look as to why the color code is yellow. And if the color code is red, it means you probably plagiarized. I'm not going to look now, but there's one of them that does not look like it's done that. And I'm not sure why that's the case. I'll have to look into that, why that one is unable to do that. I might have to take one of the students and resubmit the paper. All right, any question about any of that? So that's the grading I got done. If there's no questions about the grading, let's talk about what we're going to be doing. Let me share my screen. My computer's still a little slow because it's making the, come on, the file for the lecture. And there it is, it's at 80%, so it's only that far along. All right, today is May 21st, and we will be working on lab 9. This will be the last lab. That doesn't mean we won't have labs. You will be working on your unknown project, and some of you have not gotten started on your unknown project. Let me warn you that on average, students will need 2 weeks to get their unknown project done. Some students will need 3 or 4 weeks to get their unknown project done. And there's very few students that can finish the unknown project in about a week. I think there's been one lucky student this term who is already finished because she or he, I think it was a she, but it doesn't matter, happened to be very lucky and they just happened to get the right test and then they identified their unknown. But very few students can get the unknown project in 1 week. Most of you will require 2 weeks, and some of you 3 or even 4 weeks of work to get your unknown project done. So you need to get started on your unknown project, or you may run out of time. And if you run out of time, the unknown project will not be finished. You can submit what you got done, but you will not get 100%. The unknown project is due Saturday, June 15th. So you do have some time, but the unknown notes pages will close on the 14th. You cannot ask for any tests after the 14th. And if you ask for a test on the 14th, I will give it to you if it takes one day. And there's only a few tests that you can ask for on the 14th and get the results on the 15th. After that, you will not be able to get results. Any questions about the unknown project? You do have 1, 2, 3, 3 or 4 more weeks to get the unknown project done. And some of you need to get started because some of you have not gotten started.
I have a question.
Uh-huh.
So I already sent my second request. I forgot if I have to wait for your feedback before I request for another one. Is that correct?
To follow dichotomous logic, you have to request one test at a time.
Okay,
Okay.
that's what I thought. Thank you very much.
All right. Now, if you don't follow dichotomous logic, you still will get the results. You'll just get a penalty.
Okay.
Okay. All right. So let's talk about Lab 9, identification of gram-negative organisms. And were you the student who wanted to ask a question about the unknown project in the lecture? And I told you, I'm sorry, I'm trying to get this file uploaded. And so ask the question in the lab.
That was the other student.
Oh, no, another student. So is there another question about the unknown project before we get started on Lab 9? All right, if not, let's go with Lab 9. This will be our last real lab. Lab 9. All right, Lab 9 is the identification of gram-negative bacilli. You need to complete Lab 9 identification, meaning the worksheet, and also complete the case studies. There is some reading in Lab 9, which is normal, but not too much. Only a page on Chapter 5, biochemical tests and bacterial identification, and then a few pages on Chapter 10, methods of classifying and identifying microorganisms, maybe 3 or 4 pages. There is some supplemental material. There are video clips in this lab. The learning objections. Upon successful completion of this lab, you should be able to, 1, interpret the results for the following biochemical tests. The IMVIC reactions. IMVIC stands for the indole methyl red, Voges proscure, and the citrate test. So that's 1, 2, 3, 4 different tests. I from indole, M from methyl red, V from Voges proscure, and we usually call that the VP test. And then the I is just filled in, and then the citrate, the C from citrate test. We'll also talk about the carbohydrate fermentation test, which I think we've talked about that previously. We'll talk about the motility test and the hydrogen sulfide production test. To be able to explain how to interpret the TSI, the triple sugar iron test, or the triple sugar iron slant, the SIM test, which is sulfur indole motility test, the test of growing the cells on McConkey's agar and hectone enteric agar, and McConkey's can be abbreviated MAC, and hectone enteric is HE agar plates. And 3, use biochemical analysis to solve case studies involving gram-negative bacterial infections. So members of the family Enterobacteriaceae are all small gram-negative, non-spore forming bacilli. They are all facultative anaerobes. The Enterobacteriaceae is a very large family containing 53 genera. 24 genera have been known to cause human infections. The Enterobacteriaceae are all glucose fermenting cells. They're all oxidase negative organisms. Here are some of the genera which are normal microbiota of the intestinal tract. So we call these AKA coliforms because they grow in the colon. They can be opportunistic pathogens in that they can cause urinary tract infections, wound infections, pneumonia, and septicemia. Common genera responsible for causing opportunistic infections include Escherichia, including E. coli, Enterobacter, Klebsiella, Citrobacter, and Serratia. Several genera also cause gastroenteritis, and these are more often called just human pathogens. They include organisms in the genera Salmonella, Shigella, and then E. coli, Serovar 0157H7. So humans do not normally have Salmonella and Shigella in their intestines, and if they do get them, they are likely to get an intestinal infection causing gastroenteritis. And then E. coli is a bacteria which is normally found in your gut, but E. coli serovar 0157H7 is a human pathogen, and the E. coli with these antigens causes gastroenteritis. Any questions about the Enterobacteriaceae family? All right, the Pseudomonaceae family, including Pseudomonas aeruginosa, is similar to the family Enterobacteriaceae in that they are all gram-negative bacilli. However, the Pseudomonas are all non-glucose fermenting, oxidase-positive organisms. They can be opportunistic pathogens, particularly Pseudomonas aeruginosa, causing infections in immunosuppressed individuals, burn patients, and trauma patients. Pseudomonas aeruginosa is also a health-acquired infection, and we called that before a nosocomial infection. It's important for microbiologists to be able to identify and perform antibiotic susceptibility testing on gram-negative bacilli cultivated from a clinical specimen. In this lab, we're going to look at a few of the biochemical tests used to identify gram-negative bacilli. And here's a whole bunch of tests which can be used to identify gram-negative bacilli. We're not going to talk about all of them, okay? And here is one test where you inoculate the bacteria on this steel rod, and then you stick the steel rod through this tube so that you run 12 tests at the same time. We won't be looking at that either. We will look at some of the tests in that tube. The first test we're going to talk about is the indole test. Some organisms produce the enzyme tryptophanase that converts the amino acid tryptophan into the products indole and pyruvic acid. You then, after overnight incubation, add Kovacs reagent, which reacts to indole to produce a red color at the surface. E. coli tests positive for indole production, whereas Klebsiella pneumoniae does not. So tryptophan, in the presence of tryptophanase, that enzyme produces indole. When you have indole and Kovacs reagent added together, it's supposed to look cherry red. And as you can see, that kind of looks more purple. I'm not sure what the problem is here, if it's the photograph or not, but that's positive, and that's negative where it looks yellowish. Any question about the indole test? All right. Indole is looking at the fermentation of sugars and making indole. That's why it's called the indole test. The methyl red test is also looking at the fermentation of sugar. However, in the methyl red test, we're looking at the production of acids. And if strong acids are made in glycolysis, the MR test will turn red. If the acids are very weak, the buffer in the system can overcome it. Or if no acids are made, then the MR test will not be red, it'll turn yellow. But the point is, MR test is also looking at the fermentation of the sugar. Any question about that? All right, the MR test and the VP test, or the Voges per Skewer test, typically are ran together, and one tube is inoculated with one organism. You then incubate the tube, and you split the tube into two, one portion being used for the MR test and one portion being used for the VP test. The MR-VP broth contains peptose and glucose. It's inoculated for either 2 to 5 days. If you're running, let me see if I remember this, the VP test, you can use 2 or 5 days. If you're doing the MR test, you should run it for 5 days. If you do run it for 2 days and it's negative, you have to repeat the MR test and run it for 5 days. Because sometimes it'll be negative at 2 days, but then positive at 5 days. What you do with the MRVP broth is you split it into two. One portion is used in the MR test, and all you do is you add methyl red to the broth. If the bacteria produce acid, the methyl red will remain red, and the test will be positive. If, on the other hand, the methyl red changes color to yellow, it will be methyl red negative. And so there's a tube which is methyl red negative, and there's a tube which is methyl red positive, or MR positive. The other half of the tube you then use for the Voges per Skewer test, and you need to add Barrett's reagent A and Barrett's reagent B to it. After you add Barrett's reagent A, you're supposed to shake it for, I think, a minute, and then you add Barrett's reagent B and shake it for another minute. The minute of shaking is to add oxygen. It needs both reagents and the oxygen. And then you have to let it sit for up to about 60 minutes, looking for the production of red from the production of acetone being made in the Voges per Skewer test. If you'd see red, and it's usually only in a part of the tube, like the top part of the tube in the VP test, it's VP positive. If the tube remains yellow after 60 minutes, then it's VP negative. Okay, so if you were to look at it like in 10 minutes time, the tube would be yellow. But at 60 minutes time, the tube may be red on the top portion, and that would be VP positive. Acetone is a neutral product made, and then methyl red is an acidic product that's made from glucose metabolism. All right, any question about the MRVP test? It's actually two tests that you inoculate as one, and then you split it into two, the MR test and the VP test. If not, let's move on to the citrate utilization test. This test is used to determine whether bacteria has the ability to metabolize citrate. Citrate is the sole carbon source and energy source in the tube. And if the bacteria can metabolize citrate, citrate is a chemical found in the Krebs cycle shown here. And if citrate can be metabolized, we will then, we'll engage in the Krebs cycle, and you will make a basic product that will change the tube from green to blue, which is positive. You should see some growth, which I'm not seeing here in this one. If the tube is blue, you should see that the bacteria is actually growing and turning the tube blue. If it's blue, it's positive. If the tube is green, and usually there's no growth on a green tube, then the bacteria cannot utilize citrate, and the pH remains unchanged. This is an alkaline pH when it turns blue. So what you do is you inoculate the slant with your organism, incubate it for 1 to 2 days at 35 degrees. And if the bacteria can grow and metabolize citrate, you will get the turning blue. E. coli and Shigella can metabolize citrate, turning the tube blue. I believe Micrococcus luteus and Staphylococcus epidermidis would not change the color and it would remain green, meaning they cannot use citrate. Any questions about the citrate utilization test? All right. If not, let's go on to the PR carbohydrate metabolism test. This PR carbohydrate metabolism test has one carbohydrate, usually a sugar, which is fermented and used to determine if the bacteria have the ability to ferment that particular sugar. There is usually different sugars used in the PR carbohydrate metabolism test, including glucose, lactose, sucrose, maltose, and other sugars. If the sugar is fermented, you will get the production of an acid, which will change the color of the media from pink or reddish to yellow from the fermentation of the sugar. Sometimes with fermentation, you can also get gas production. So this is called A for acid being produced in the fermentation of the sugar. This is called acid and gas because the acid is produced, the sugar is fermented, changing the color from pink or reddish to yellow. And then gas is produced in that inverted Durham tube. So we can see a bubble, and that's called AG. Any question about PR carbohydrate metabolism? So in the unknown project, you will be doing, well, all of you will do lactose fermentation, and you will not find that under lactose fermentation. You will find it under PR carbohydrate, probably metabolism, but maybe fermentation. Any question about that? The PR stands for phenol red. It is the pH indicator, which turns pink or reddish when the pH is pH 7 or less. And if it's an acidic, it'll turn yellow. All right, if not, let's talk about the motility medium test. You inoculate a tube for the motility medium test to look to see if the bacteria possess flagella, and that means they're motile and they can swim. And the motility medium is a semi-solid medium, a very low agar concentration, so the bacteria can swim through it. If it was high agar concentration, which is normal, the bacteria could not swim through it because there's just too much agar. The bacteria can swim through it. But if it's low agar, I think it's 0.4%, but maybe I'm wrong. Maybe it's 0.1%. And you stab inoculated. If the bacteria only grow in the stab, shown here, the bacteria are not motile and they will only grow in the stab. If, on the other hand, the bacteria will be everywhere, and there you can barely make out the stab, and the tube is cloudy from the bacteria swimming everywhere and then growing everywhere. And that would be a very motile bacteria. You can have a slightly motile bacteria, in which case you won't see the bacteria growing everywhere. You'll have your stab, which will be here, and then the bacteria will be flaring. I should only do it on one side. Flaring out from the stab. And it doesn't have to be the same pattern. So I'll do it like that. And here we'll do it like that. Where the bacteria flares out, grows out a little bit from the stab. And that's called flaring, where the bacteria, let's go right there, grows out a little bit from the stab. And I don't have a real picture of that, but bacteria that are slightly motile will not swim everywhere because it's only slightly motile. It only swims someplace, and it looks like a flare like that. So if the bacteria make the media turbid looking or it flared, then the bacteria has a flagella and it's mobile. And if it only grows in the stab, the bacteria is non-motile. Let's talk about the hydrogen sulfide test. With the hydrogen sulfide test, we're looking for the production of hydrogen sulfide. You can run this test in at least 3 different media. You can run it in the triple sugar iron, also called the TSI agar slant. You can run it in the SIMS test. That's a sulfide indole motility deep test. You can test it on the hectone enteric agar. In the production of hydrogen sulfide, the hydrogen sulfide, if it's produced, reacts with something in the media that forms a black precipitate. And then you'll see black showing up in the media. Citrobacter, Salmonella, and Proteus species all produce hydrogen sulfide, and they will turn the media black. Micrococcus luteus will be hydrogen sulfide negative, and the media will not be black. Usually you see black at the bottom of the tube because hydrogen sulfide is produced anaerobically. And so at the top of the tube is not black because anaerobic respiration is not occurring. Probably aerobic respiration is occurring. So usually you see the hydrogen sulfide in the bottom of the tube. Any question about hydrogen sulfide? Now this is showing you, I think that's a SIMS test showing you here. I'll show you that again. This is turning black in the triple sugar iron test. Once again, only the bottom of the tube's black. The top portion is not. SIMS for sure. black on the bottom. You can also test the hydrogen sulfide production on the HE plate, ectone enteric agar. If the bacteria produce hydrogen sulfide, then the colonies will look black on the HE plate. If the bacteria cannot produce hydrogen sulfide, then the bacteria will be their normal color, which in this case looks white. Any question about that? All right, that's the hydrogen sulfide production test. Let's talk about the triple sugar iron slant test, also called the TSI test. What you do in the TSI test is you take your needle and get bacteria on the tip of the needle, then you inoculate the butt of the tube. And you should go down 2/3. That's only going down halfway. So you should go down to about here. You don't want to go to the very bottom because you don't want to damage the needle. But you do want to go about 2/3 of the way down. And that will put bacteria low down in the butt of the TSI tube so that the bacteria can grow anaerobically with little or with no oxygen. And then you pull out the needle, and then once again you get bacteria on the tip of the needle, and then you streak the slant of the TSI tube so that the bacteria can grow aerobically. So we're looking here at growth aerobically, and there we're looking at growth anaerobically. And then you take a look at the tube to see what happens. This test is very complex. Let me mention that the tube has only 0.1% glucose, but 10 times higher lactose and sucrose, meaning 1% lactose and 1% sucrose. It has the pH indicator in it, phenol red, which changes color depending on the pH. There's peptone for a source of nitrogen, and it may be metabolized. And then it has sodium thiosulfate, sulfate, and ferrous sulfate to look for the production of hydrogen sulfide. So if hydrogen sulfide is produced in the bottom of the tube will turn black in the bottom. If there's no black, then no hydrogen sulfide is produced. I'll come back to this tube. Let me talk about this tube because this one's very easy to discuss. We note here that it's yellow on the slant and yellow in the butt. So this we record as A for the slant, acid for the slant, A for acid in the butt. So A over A, whenever it's yellow and yellow, then we know glucose is fermented and one other sugar or both other sugars. We don't know if it's one or other, but because it's yellow and yellow, we know that both glucose is fermented and at least one or both lactose and sucrose. We know in this case the gas is produced because we have a bubble right here and then cracking of the agar right there. So gas is produced. No hydrogen sulfide is produced because the tube, especially the bottom, is not black. Any question about that? In tube 3, we have, I'm going to put that negative, but you can put K. K is for alkaline, meaning it turned pink. It's alkaline because the proteins are metabolized. If you metabolize proteins, you release ammonia. Ammonia is a base, and that'll change the pH color to pink, and that actually is pink. So that's why it's called the K. But you can put that as negative also. And then the butt is yellow. So we say A is going on. What this means when the butt is yellow and the slant is pink, then we know that only glucose is fermented. The reason why is glucose is at low concentration and it is quickly metabolized aerobically, but it is metabolized slower anaerobically. And so the slant is pink because all of the glucose was fermented. And if we had looked at this tube early on, this would have been yellow on the slant. But after the time scan, like a day or two of the tube growing, the bacteria, rather than dying, will start, once all the glucose is fermented, will start metabolizing the protein and then will change what was yellow. We call it reverting what was yellow to a pink color as the protein is metabolized aerobically, producing a base, changing the color from yellow to pink. So in the time span of this experiment, this will be pink, and we say negative over A, and that means only glucose fermented because the glucose is fermented slower anaerobically, and so the butt is yellow, the slant is pink. And I know that's complex. I'm not sure how to tell you how to remember this, but you should try to remember this. There's no black here, so no hydrogen sulfide is being produced. And I don't know if I mentioned the reason why the glucose gets used up is because it's only at 0.1%, whereas here the slant remains yellow because it's fermenting the sugar, either sucrose or lactose, which is higher, 10 times higher than the glucose. On tube 2, because the slant is pink and the butt is black, we know that only glucose is fermented. If we had looked at the tube earlier, that would have been yellow, but because the glucose got all used up, the bacteria, rather than dying, start metabolizing the protein, producing a base, changing the color of the slant from yellow to pink. Because the butt is black, this happens only in anaerobic respiration. And if anaerobic respiration is occurring, then we know that, meaning if it's black, we also know that it's yellow. Now, in this case, you can't see any yellow here. And so you just have to know that if it's black, there's also yellow. If we'd looked at this tube much earlier, it may have been only black in the center, and then the edge of the butt would have been yellow. So this is put K for the slant or negative, and then A for acid being occurring in the butt. No gas being produced, but in this case, there is hydrogen sulfide being produced, and that's why it's black. And then tube 1 is negative over negative, or K over K, meaning alkaline over alkaline. No sugars are fermented, no yellow anyplace. No gas is produced, no breaking or lifting of the auger, and then no hydrogen sulfide is not black. Any questions about the TSI test? If not, let's talk about the sulfide indole motility test. This test is running 3 tests at one time. We're looking at the production of sulfide, hydrogen sulfide being produced. We're looking to see if the bacteria is motile. We're looking to see sulfur indole motility and the production of indole. So 3 tests are being run at the same time in the SEMS test. What you do is you stab inoculate the bacteria. You can see the stab inoculation right there and right there. You then incubate overnight, pull out the tube, and look at the tube. If the tube, you first, before looking at the tube, You add Kovacs reagent. If Kovacs reagent turns pink, then it's indole positive. If the Kovacs reagent remains yellow, then it's indole negative. You look to see if hydrogen sulfide is produced in the bottom of the tube. If hydrogen sulfide is produced, the bottom of the tube will turn black. The very top may not be black. That's actually Kovacs reagent on top, but the very top may not be black, and that's because the hydrogen sulfide is produced anaerobically. So the very top of the tube may not be black. And then you look at the motility. If the bacteria only grow in the stab, then the bacteria are non-motile. So that one there, the bacteria is only growing in the stab. The same with that one, although that one's a little more difficult to see. And it looks like the bacteria are growing more rapidly here than there. But this one, you can note the bacteria is flaring out there, and it's a real big flare over here. So this means the bacteria is motile. It is swimming through the Sims media. And so the bacteria do possess a flagella. Any questions about the Sims test? All right. We're also going to be talking about the McConkey's auger test and the HE or hecton-enteric auger test. If you grow the bacteria on McConkey's auger, if the bacteria can grow, the bacteria are likely to be gram-negative because McConkey's auger selects against gram-positive cells. McConkey's auger has bile salts in it, and I'm going to get it. Just let me get a crystal violet in it, which is why the gram-positive cells do not grow. Crystal violet gets stuck in the cell wall, preventing the cells from growing. Gram-positive cells don't like growing around bile salts. Bile salts are found in your gut, and gram-negatives are normally growing in your gut. So they typically grow on McConkey's. So if there's growth, it's likely to be a gram-negative. If there's no growth, it's likely to be a gram-positive. And that's true about 95% of bacteria. There are a few bacteria that do not do as expected, but most do. And then on McConkey's auger, you can also look to see if the bacteria ferment the sugar lactose and then differentiate them on lactose fermentation. If the bacteria can ferment lactose and grow on McConkey's, the bacteria will look pinkish, both in the color of the colonies. Let me blow that up. That's not up. As well as in the agar around where the bacteria is growing. If the bacteria cannot ferment the sugar lactose, the bacteria will be their normal color, which in this case, the bacteria are normally white or off-white. For the hecton-enteric auger, if the bacteria can grow on the plate, the bacteria is likely to be gram-negative because the bacteria select against gram-positive. So gram-positives tend not to grow on the hecton-enteric auger. We can also differentiate the bacteria from their hydrogen sulfide production. If the bacteria do not produce hydrogen sulfide, there will be their normal color. If they do produce hydrogen sulfide, they will be black. You can also differentiate the growth on the hecton-enteric auger by the fermentation of the sugar lactose. If the bacteria does not ferment the sugar lactose, then the bacteria will be their normal color, which in this case looks whitish, maybe off-white for that one. But if the bacteria do ferment the sugar lactose, they will turn salmon color. That's a yellow-orange color on hectoenteric agar. And that's only if they can grow. Will they turn the plate yellow-orange or salmon color? If the bacteria cannot grow, you cannot run any of these tests, either the lactose fermentation or the hydrogen sulfide production. Any question about McConkey's agar or HE plate? All right, lab 9. While you're not going to be physically performing any of these tests, because everything we do will be done online, you do need to know how to perform and interpret these tests, and you will be held responsible for that, and there will be quiz questions on that. We have some video clips for you to watch on carbohydrate fermentation, a video clip on the SIMS test, a video clip on the MRVP test, and that's actually 2 tests starting as one, and then a video on the TSI, triple sugar iron test, and then a video on McConkey's agar. You'll also need to view some of the website microbuzz. I'm not sure why that's so small there. Come on, mouse. Uh-oh. Looks like they changed this link. I might have to find out where microbuzz is, and let me look at the worksheet and see if the link is there. Nope, it's not here. Let me try that again. Immediately. This looks like more of their college information. Make sure I spell that right. B-U-G-Z. Nope. Page not found. That was there on the link. All right, I'm gonna have to do a search, I think, with Google to try and find microbuzz because it's not coming from the own college search. You'll have to do your best on this lab until I get a link on microbuzz. I'll try and get a new link on microbuzz. I'm hoping they still have it available. We just had to find where they moved it to. But this is a very recent change because I worked on that just last term. It was fine. Supposedly you should look at microbuzz and you need to click on some links about the tests you need to complete for step 1 in the worksheet to lab 9. And then you have the case studies below to work with. Let's go through the worksheet briefly. So to fill in this test, you need to look up some information about these tests: the indole, the methylred, the Voges Proscure, the VP test, the citrate test, carbohydrate fermentation, TSI, and the SIMS test. All of those you got. Actually, everything you can get from the lab, so you don't really need the microbuzz website to answer these questions. But the first one has been filled in for you. In the oxidase test, the result of a positive is the cell or spot turns blue. The negative is no color change. So you need to say what the positive result is for each of these tests: the indole, methylred, the VP, citrate, and give the negative result. You will get points for both the positive result and the negative result, so you do need to put in the negative result. It'll probably be 0.1 point for each correct answer. And then complete the worksheet of the questions under 2. So which reagent must be added to detect the presence of indole? If indole is present, the test will show what color. So state the reagent that's added in the indole test and what color will it be if indole is present. 2B, methyl red is a pH indicator that remains what color under acidic conditions, and methyl red pH indicator is used in which test? For 2B2, I guess. For 2C1, the what test uses Barrett's A and Barrett's B reagents to detect the presence of acetone, a neutral product formed from glucose metabolism. If acetone is present, the test will show what color? 2D, Simmons citrate is used as a pH indicator. In what test? And turns blue under bold or acidic conditions. You need to write bold or acidic right there. E, what is the pH indicator used to detect PR, carbohydrate fermentation? So what's the pH indicator name? And it is yellow under either bold or, acidic or basic. You can either bold that, I guess you could underline it or write it right there. Bacteria that possess this structure test positive for motility. So name the cell structure. 2G, 3 organisms that produce hydrogen sulfide. Name 3 organisms. You should name a species. The exception would be if the entire genus or family produces hydrogen sulfide, then you could give the genus or family. For example, you cannot say family Enterobacteriaceae because not all members of the family produce hydrogen sulfide. 2H, if an organism ferments glucose but not lactose or sucrose, the TI slant will be what color, and the deep butt will be what color. And then 2I, state the 3 tests for which SIM is used for, meaning what does SIM stand for? 2J, MAC and HE agar plates are both what? In that they inhibit the growth. And then bold or right one, gram positive or gram negative bacteria. And they are also what? And they separate various bacteria based on visual differences from sugar fermentation. And then 3, complete the case studies identification below. Use this table to identify the bacteria in your case studies. Do not use this table to identify your own known bacteria or any bacteria in any other lab. Only use this table as a dichotomous key for the bacteria in the case studies in this lab. And this table got split. Sorry about that. There's top part of the table and there's the bottom part of the table, one row. For this table, you can put in the information about case study 1, whether it was oxidase positive or negative, whether it ferments glucose or not, whether it ferments lactose or not, what's the results of the TSI. And here, this thing would be to be something like negative over negative, meaning slant over but you could write it all out. You just need to expand that. Then hydrogen sulfide production, that's either plus or minus. Motility, plus or minus. The Undol test, the MR test, which these are both plus and minus. The VP test, also plus and minus. And then the citrate utilization test. So do that for case study 1, what the results are. Then use this data compared to this table here to determine what the species is. And for case study 1, put the species causing the case study here. And do that for case study 2, 3, 4, and 5. So write the answer on the worksheet. For looking at the case study, you need to go to the lab manual. Case study 1, a prison inmate is brought into the ER room with an open wound, and this prisoner is known for self-inflicting. A routine culture and sensitivity test is ordered. The appropriate transport media is used for specimen collection and inoculation of the prescribed media. So here we have the bacteria from the patient's infection growing on a blood agar plate, and you can look at the hemolytic pattern from that. We then took some of these cells and grew it on a McConkey's plate, and you can tell that there's growth on the plate, and we can see a color. So you need to know what that means, the purple color on McConkey's. And then we have the gram stain here. I suggest what you do is you blow that way up to look at it better and try and determine what the shape of the organism is, right there and right there and right there and right there. And maybe the gram stain, whether it's gram negative or gram positive. I knew there are two things. So you can get the shape of the bacteria and the gram stain of the bacteria. And then we have some more tests. We did a rapid test to determine that the cells were catalase positive and oxidase negative. And then the isolated colonies are used in the biochemical test below. And here's the results of day 2. Now this page got split, but this tells you that's the Sims tube right there. Let me see if I shrink this down a little better. There we go. So you can see the results of the Sims tube there. This is the results of the MR test. This is the results of the VP test, the results of the Simmons citrate test, and then the results of the triple sugar iron test. Any question about, that was supposed to be case study 1. Oh, okay. Sorry, there should be more space between case study 1 and case study 2. And then here's case study 2. So that's part of that table there. That's part of case study 1. Mikey talks his mom into buying a pet turtle. He does well with changing the water, but he's not diligent about washing his hands. About 2 weeks later, Mikey develops diarrhea, nausea, and fever. His pediatrician orders a fecal sample and routine culture testing. Here we see the bacteria growing on McConkey's agar with the arrow pointing at a bacteria of interest, and the arrow showing the bacteria of interest on an HE plate. The bacteria are catalase positive and oxidase negative. So what does that mean? And then here we have the bacteria in Sims tube, the bacteria in MR test, the bacteria in a VP test, bacteria in a Simmons citrate test, and the bacteria on the TSI test. So fill in that information in the worksheet here. And then look at the key to identify the species and write the species here. Any question about how to do the case studies? All right, I'll let you read about the other case studies. I will be here at 8 till 8 o'clock to help you with any questions. Please use me if you need me. I'm most grateful to help you. Any questions? If not, I'll turn it over and you guys can start working on the lab. I will be here until 8 o'clock or the last student logs out.